Hey y'all, welcome back to another edition of Purple Bear Biology. In this episode, we're going to focus on 4 primary types of macromolecules that have a dramatic impact on our body's anatomy and physiology. We're going to discuss carbohydrates, nucleic acids, proteins, and lipids. Let's jump right into the slides and take a look at some of these cool compounds. Let's start with what a macromolecule is. When we talk about 4 primary types of organic macromolecules in our body, we are really describing compounds made of monomers. I like to think of it like Legos, where each monomer is an individual Lego brick, and the macromolecule is a stack of connecting bricks. Most organic macromolecules are polymers, meaning they have a series of the same or similar repeating monomers. What connects the monomers together to make polymers? Well, bonds, of course. The bonds are different depending on the macromolecule. So let's start with one that you are pretty familiar with. Carbohydrates, or as you probably know them, sugars. Different types of sugar molecules exist due to different combinations that are created with sugar monomers. Cool fact. To combine individual monomers of carbs, called monosaccharides, a reaction called dehydration synthesis removes water and links the 2 monomers together with covalent bonds to an oxygen atom. The reaction can be reversed to break them apart using energy in water. Breaking sugars down this way is called hydrolysis. So what is the primary function of carbohydrates in our body? They serve as an energy source. The sugars can be broken down, and as they are broken apart, energy can be harvested from the reaction. Carbohydrates can also be found on the surface of our cells' plasma membranes. They act a lot like flags on a ship to show their identification. This is one way that our immune system can identify self cells versus cells from an infection. While we are talking about sugar's usefulness, we can move on to nucleic acids. Typically, when we think of nucleic acids, we think of things like RNA or DNA. Both DNA and RNA contain sugar repeating units that make up the backbone of the structure. DNA serves as the genetic instructions for our cells, and RNA is used to take that information and turn it into structures like proteins. While the two are very similar, there are some notable differences between DNA and RNA. First, the sugar monomer that makes them up is different. Additionally, DNA is double-stranded, and this makes it more stable than RNA. You may have also noted that in addition to sugar, nucleotides have a nitrogenous base attached. DNA and RNA share 3 of the same nitrogenous bases, but DNA has a thymine while RNA has a uracil. These nitrogenous bases are the location of interaction between DNA strands and are used as a coded message that gets transcribed to RNA and then translated into protein. All of the diversity that we see in and around us can be tracked back to these 4 nucleotides. But how does a message so complicated come from a series of repeating chemical compounds? Well, it's actually super cool. See, nucleotides have complementary nitrogenous base pairings, kind of like puzzle pieces that fit together. Guanine always binds to cytosine, and adenine always binds to thymine, or to uracil if this is RNA. So, how can we use this information to build an RNA template from our DNA? Well, let's take a look at this strand of DNA. If I look here, I can see that the first nucleotide in here has an adenine. That tells me that the next nucleotide on the opposite strand should be a thymine, because A's always bind with T's. Except in this case, we're not going from DNA to DNA, we're going from DNA to RNA. So, adenine will bind to uracil instead of a thymine. The next nucleotide is a cytosine. We know that cytosines always bind to guanines, so C's bind to G's. So if I were to start creating this template down here based on this individual sequence, what we could see is that it would be a U, a G, a G, a U, a U, a U, a G, a G, a C, a U, a C, and then an A. So let's see if I got that right. Look there. I can predict what the RNA should be based on what the DNA is, because of complementary pairing. This is called transcription and takes place inside of the nucleus of a cell. Now, if I want to turn this into a useful protein, I need to perform translation. Translation is a little trickier, but not by much. What must happen next is that RNA is decoded by other RNA structures. This special decoder is called tRNA, and it reads RNA messages in sets of 3 nucleotides called codons. Each codon is associated with a particular amino acid chemical. To figure out which one, you must use the nitrogenous bases as a key to this amino acid chart. Let's look at the first example. UGG is the first codon in this RNA sequence. So I can use this table by starting with my first letter, which is U, finding the box that correlates to the second letter, which is G, and then the third letter in this box, which is also G. Tells me that the amino acid that I need is abbreviated as TRP. That means the first amino acid in the sequence would be TRP. Let's look at another example, the second codon in this sequence. Notice that it's U, U, U. So my first letter is U, my second letter is U. Now I need to find my third letter, which is depicted here as U, and that tells me that the second amino acid in this sequence should be abbreviated as PHE. As these get translated, they get bonded together with covalent bonds called peptide bonds. The final product is an amino acid sequence that will chemically interact to form a functional shape we call proteins. We will continue our conversation about proteins in a second, but I want to discuss one more nucleic acid that is indescribably important, ATP. Adenine triphosphate, aka ATP, is a nucleic acid and an example of an energy carrier. The molecule is built with the same sugar found in RNA and the same nitrogenous bases found in both DNA and RNA. ATP is incredibly important because during metabolic breakdown of carbohydrates, energy gets invested into making a covalent bond between the phosphate chain. This energy can be harvested later and used to perform essential processes throughout our cells. Okay, let's move on and return our attention to proteins that are getting built. Proteins, as mentioned before, are chains of amino acids connected together with covalent peptide bonds. Proteins function to speed up chemical reactions, provide structural support, enable cell-to-cell communication, enable muscles to contract, and so much more. There are 3 different types of proteins involved in attaching one cell to another in the process of forming tissues. Tight junctions act kind of like zippers, weaving the membranes together to prevent anything from passing between the cells. This type of junction are typically seen in areas that are watertight, like the skin. Desmosomes act like buttons on a coat. They keep the 2 membranes together, but allow fluid to pass between the cells. These connections are particularly strong and can be found in areas that receive stress due to movement, like skeletal muscle, the skin, and the heart. The last connection is called a gap junction. These connect one cell's intracellular fluid to another cell and allow tissues to coordinate stimuli. This type of junction is incredibly important in areas that require close coordination, like cardiac cellular contraction. Proteins also help to speed up chemical reactions. Proteins that do this are referred to as enzymes. Enzymes can build compounds in reactions called anabolic reactions or break compounds down in reactions called catabolic reactions. The most important variable that influences protein function, including enzymes, is their shape. The shape of a protein determines how it interacts with the environment. If the environment changes the shape of a protein, then it may reduce or eliminate the function of that protein. Preserving protein function is one of the primary reasons our bodies have homeostatic set points. Temperature and pH both affect protein shape, and thus are tightly regulated for homeostasis. The environment, of course, is not the only way that proteins may be misshapen. Don't forget that proteins are built from DNA. Changes in a single nucleotide can result in life-threatening diseases due to malfunctioning proteins. For example, sickle cell anemia is a genetic disease that causes hemoglobin proteins in red blood cells to clump up and form rods. These rods change the shape of the red blood cell, making it less effective at traveling through the body. Sickle cell disease is caused by a single nucleotide change. A single adenine switches and becomes a thymine, resulting in a different amino acid in the sequence and creates this life-threatening disease. While the effects of the disease are devastating, the pathology of it is fascinating. Proteins also serve as a vital function in transport across the cellular membrane. But we are going to talk all about protein transport and osmosis in another episode. So let's switch gears and talk about our last macromolecule, lipids. You're probably familiar with lipids from the perspective of cooking with fats and oils. Lipids, in the form of fatty acids, are long chains of carbon atoms that can be utilized as an energy source just like carbohydrates. You have probably heard that saturated fats are bad for you. The term saturated is referring to the number of bonds carbons in the chain are forming. When two carbon atoms share two electrons, this is called a double bond, and it causes the lipid to bend. Saturated fats do not have this double bond. While this may seem like a simple thing, we have just seen how shape can influence how our bodies respond in a dynamic way. Saturated fats can lead to higher levels of bad cholesterol in the blood, which in turn can lead to plaque buildup in the walls of arteries. When unsaturated fats are substituted in the diet in place of saturated ones, bad cholesterol levels can drop as much as 10%. Another really important lipid is triglycerides. Triglycerides are lipids that can store energy for future use. The body stores this energy by creating bonds between a glycerol molecule and 3 fatty acids. While we can find triglycerides in the blood when they are being mobilized for cellular metabolism, they are also stored in large quantities in fat cells. Believe it or not, these lipids actually provide more energy than carbohydrates when broken down into their fatty acid chains. Steroids are also lipids. Most people have heard of steroids. Some probably recall it from medication a doctor provided, while others may have heard of individuals abusing steroids to gain muscle mass. Steroids themselves are a specialized group of hormones that can be divided into 2 primary categories: corticosteroids and sex steroids. Our body naturally makes corticosteroids to help reduce inflammation or alleviate an immune system response. This is similar to the synthetic ones you may receive when you go to your doctor and you're suffering from something like asthma or rheumatoid arthritis. Cortisol is a corticosteroid involved in the body's response to stress and can have multiple effects all around the body. Anabolic steroids primarily have the reputation from bodybuilders and athletes trying to increase muscle mass quickly. While misuse of synthetic anabolic steroids can lead to terrible complications, especially after long-term use, the steroid actually mimics what is needed for individuals to grow from childhood into adulthood. These steroids are mainly used in medications to fight effects of chronic wasting disease, late-stage AIDS, and even to treat bone disease like osteoporosis. The last lipid in our adventure is a phospholipid, and they are the primary component of the plasma membrane. Lipids generally are long nonpolar molecules, but in phospholipids, these long nonpolar chains are attached to polar phosphate heads. Because they have polar heads and nonpolar tails, the heads orient themselves facing water's environment, and the tails always face each other. Your plasma membrane is a double layer of phospholipids, and it's the nonpolar tails that prevent polar molecules from moving freely in and out of the cell. These structures are incredibly important for the cell to regulate what enters and leaves in conjunction with proteins embedded in between them. Wow! Isn't the chemistry of the body mind-blowing? It's pretty amazing to think that our bodies represent highly organized chemical compounds that aid us in everything from providing energy to cellular function. I hope you found this introduction to macromolecules helpful. If you have comments, be sure to leave them in the comment section below. And don't forget to hit the like and subscribe button if you enjoyed the video so that you can continue to see additional videos I release. See y'all next time, and thanks for stopping by.